Neurofibromatosis is an autosomal dominant disorder. Although 50% of the people with this condition can result from sporadic new mutation in the gene with no family history of the disorder. Neurofibromatosis affects all neural crest origin cells. The tumor will begin in the myelin sheath of a neuron. The spread of tumors causes other deformities, and it is highly differentiated between cases. If you look at the diagram below, you'll see that it affects myocytes, melanocytes, adipocytes, osteocytes, neurons, Schwann cells. It's just amazing how many different neural crest cells that this condition affects. There are two types of neurofibromatosis, and the difference is where it lands on the gene. Type 1, or NF1, is also known as von Recklinghausen disease. It is the most common type of neurofibromatosis, representing 90% of all cases. And unfortunately, it's reasonably common. Incidents are 1 in 3 to 4,000 worldwide and affects males equally as females. Type 2 neurofibromatosis is rare and only represents about 10% of all cases. The incidence is 1 in 40 to 50,000, and it also affects males equally as females. So what is happening with neurofibromatosis? Well, it is tumor suppressor gene mutation in both type 1 and type 2. So if you remember way back from basic biology, all cells have a life cycle. They have the cell cycle where they grow, where the DNA is duplicated, where the DNA splits, and then you have mitosis. And before mitosis, you have interphase where the cell is doing its work. And there's several places throughout the growth of a cell that are checkpoints, and you can see in the diagram we're representing checkpoint 1 and checkpoint 2. Well, these checkpoints are monitored by tumor suppressor genes, where the gene says, okay, you're growing and you're growing at a good rate. I'm going to let you continue on. If it decides that the cell is growing too fast, as in the case of a cancer cell, it'll stop the cell at that point and not allow it to continue to grow. Then the next checkpoint is where DNA synthesis and division has occurred. So it checks the DNA duplication and it decides, this DNA, it looks good, so I'll let it move on. Or it may say, this replication of the DNA is not quite right, so I'm going to stop it right here and not let that cell undergo cell division. Well, in the case of neurofibromatosis, they don't have normally functioning tumor suppressor genes. The tumor suppressor gene that is in question is called neurofibromin, and there's a mutation with that gene, and it causes ineffective checkpoints. And if you don't have an effective checkpoint, it's going to lead to uncontrolled cell division and a tumor. Neurofibromatosis type 1 early signs are cafe au lait spots and freckles in the axilla or groin area, and these are noticed mostly in early childhood. Adult signs and symptoms of neurofibromatosis 1 are neurofibromas on the neurons, and they are mostly cutaneous. You can see on this individual the photo on the right of this young lady where she has fibromas all over her face, and that's just a picture of her face. Her entire body is covered with these fibromas that represent like little bumps. These are tumors of the myelin sheath. Now, for the most part, these are benign, but about 5% of these types of tumors can become cancerous. Adult signs and symptoms of neurofibromatosis include Leck nodules, which are brown spots visible in the iris of the eyes. These spots, or Leck nodules, do not interfere with the individual's vision. However, NF1 is also subject to optic gliomas. They may or may not always cause any kind of partial or complete loss of vision, but they are more likely to cause the vision problems where LEC nodules will not cause any. Adult signs and symptoms of NF1 continued are high blood pressure, scoliosis, and osteoporosis. If you look at that x-ray on the right, you'll see the individual has multiple tumors. It's the tumors that you can see on the skin are projecting through the x-ray, but neurons travel throughout the entire body. So what you see on somebody's skin, that's just the surface. And there will be those same type of nodules on the myelin sheaths of any kind of peripheral neuron running through the interior of the body. NF1 diagnosis in a summary is cafe au lait spots, 2 or more neurofibromas, or a plexiform, which is a group of neurofibromas, freckling of the axilla or groin region, optic gliomas, thinning of long bones of the cortex, because osteocytes are also neurocrest cells or origin cells, LEC nodules that we talked about. A first degree with the condition is a no-brainer, and genetic tests are very easy to determine whether or not there is neurofibromatosis involved. Neurofibromatosis 2 is also autosomal dominant. It is rare. The frequency is 1 in 40,000 to 1 in 50,000. It's most commonly associated with cranial nerve 8 schwannomas, which is a neuroma of the vestibular cochlear nerve, and multiple meningiomas. Signs and symptoms of neurofibromatosis typically appear during adolescence or in the early 20s. The symptoms are migraines, and I might say a sudden onset of migraines, hearing loss, tinnitus, and problems with balance. A vestibular schwannoma associated with NF2 is the number one pathology associated with this condition. It's also called an acoustic neuroma, and it only affects cranial nerve 8. Multiple meningiomas associated with NF2 are typically tumors of the meninges, brain, and spinal cord. About 90% of these are benign, but 10% could become cancerous. The way to diagnose NF2 is through an MRI looking for the tumor of the cranial nerve 8. You can also do a genetic test. And one of the differential diagnoses is anything else that could cause a tumor in that area, and Meniers disease, which we are going to talk about next. Meniers disease is a disorder of the inner ear. It's also known as endolymphatic hydrops. Meniers disease is a buildup of endolymph in the labyrinth of the inner ear. It increases the hydrolytic pressure within the ear, making it feel very full or like you have water in your ears. Meniers disease etiology is that the cause is totally unknown, idiopathic. It can occur at any age, but it typically affects individuals between the ages of 40 to 60. And about 1% prevalence in the U.S. of the U.S. population. Familial predisposition may increase the chances of someone having Meniers disease, but it doesn't mean you will get it. 3 to 8 times more common in women than men, and it primarily affects Caucasians. I've actually had 3 patients with Meniers disease in my years of practice, and all 3 of them were old white men. Meniers disease symptoms are dizziness, specifically vertigo, which is associated with nausea and vomiting. So the symptom of nausea and vomiting is not specifically due to Meniers disease, it's due to the vertigo associated with Meniers disease. It also has ringing in the ears, tinnitus, hearing loss, and a feeling of fullness or congestion in the ear, known as oral fullness. Now, this is very similar to the symptoms of neurofibromatosis 2. The main difference is neurofibromatosis 2 doesn't have this feeling of fullness or congestion in the ear, and Meniers disease does not typically have migraines associated with it. Now, somebody who has Meniers disease could also have migraines, but they probably would have had them before they had the Meniers disease symptoms. But it is a differential diagnosis between those two conditions, Meniers and NF2. Meniers disease diagnosis continue. For someone to be diagnosed with this condition, they would have to have 2 or more episodes of vertigo lasting at least 20 minutes each. They would have tinnitus, ringing in the ears, temporary hearing loss that would be confirmed by audiometry exam, oral fullness, which is the feeling of congestion or like you always have water in your ear, confirmed by otoscopic exam, and an MRI or a CAT scan to rule out a tumor that would be associated with NF2, Meniers disease, to rule out a schwannoma. Meniers disease treatment. There's no medical cure. There's no specific wonderful medication that'll fix it. Medication is given to help with the dizziness or the vertigo. There will be some dietary restrictions trying to reduce body fluids overall by reducing salt in the diet combined with diuretics. The whole goal is to lower extracellular fluid, lower blood pressure, lower the amount of volume of endolymph. And the more radical treatment is surgery to relieve the dizziness, where they actually would cut the vestibular cochlear nerve, which is pretty radical, or they might actually cut the labyrinth, a labyrinectomy. Meniers disease prognosis is it's not fatal, and individuals will often have attacks that come and go. The biggest problem and biggest concern with Meniers disease is that the dizziness associated with the condition can come on suddenly, and they would have a sudden loss of balance and just fall down. These are called drop attacks, and these sudden falling is where someone could actually have a fatal injury hitting their head. So that's the most concern with Meniers disease, are drop attacks. Meniers disease attacks about twice a year, and that's average. The remission rate is 50% will have full remission within 2 years, and over 70% will have full remission in 8 years, meaning that it doesn't come back. The medical model manages the symptoms with medication, particularly treating the dizziness. However, chiropractic adjustments are extremely effective in eliminating the symptoms. I encourage you to watch the video I put up about Meniers disease and chiropractic care. Now we have moved on to Prions disease. There are several types of Prions disease. A couple of them are Creutzfeldt-Jacobs disease, and it can be contracted either sporadically, unknown, or iatrogenic, medically induced. And then there's variant Creutzfeldt-Jakob disease, which is contracted via eating contaminated meat. And then there's another Prions disease, fatal familial insomnia, which is genetic. And then Kuru in humans, that occurs only in New Guinea, or that's the only place it's been discovered or isolated, and it's a result of cannibalism. So what are prions? They are abnormally folded proteins. When proteins are initially formed in the ribosome, they're linear, and then they go into their folding process. The folds can be secondary, tertiary, and globular. The size or folding of the proteins determines its function. When these prions are abnormally folded, they actually become rod-shaped and very sharp and can poke holes in CNS tissue. So, what do prions do? Just what I mentioned before. They poke holes in brain tissue. In some of the conditions of Prions disease, specifically Creutzfeldt-Jakob, it can develop a sponge-like texture of the CNS tissue, and that becomes what's called spongiform of the CNS tissue. If you look on the left, you'll see CNS tissue with all these white circles, and that's where the prions have actually punctured this tissue, and there's a hole there, and it's dead. And on the right, you can see normal tissue. This occurs with most of the Prions disease. So what is Prions disease? It is a degenerative neurological disorder that can lead to focal damage of central nervous system tissue, causing the death of neurons and glia cells. It affects the cerebral cortex, basal ganglia, the thalamus, the cerebellum, the brainstem, the spinal cord. There is no CNS tissue that prions cannot affect. The signs and symptoms of Prions disease, with the exception of fatal familial insomnia, is a rapid decrease of mental function, dementia, and abnormal movement, particularly if it gets the basal ganglia. And the mortality is fatal. In fact, all Prions diseases are fatal. Here's a couple types I'm going to review. Sporadic CJD is the most common type. 85% of all cases of Creutzfeldt-Jakob disease is the spontaneous conversion of the protein shape from its normal shape to a rod-shaped prion. The next one is iatrogenic, and it is contagious in that it's contaminated tissues that are accidentally transmitted surgically. For example, if it's for a graft, specifically corneal implants, or if it's medications, specifically growth hormone medications that are contaminated. This can pass on Prions disease. Variant Creutzfeldt-Jakob disease is by contracting or consuming an infected meat. Fatal familial insomnia is genetic, where there's a mutation in the gene that codes for the prion protein, which causes the misfolding. And Kuru is from consuming infected human brains or contacting a wound of an infected person. Sporadic CJD statistics are the onset typically occurs over the age of 40. The average age is 60 to 68, but it can occur at any age, so we have a range from 16 to 82. It affects males the same as females. It is extremely rare, 1 in a million, and it is fatal, as all Prions diseases are fatal. The average survival is by about the onset of symptoms, the individual will probably die within 7 months. So I mentioned before, iatrogenic CJD is transmitted medically. Surgically, the most common way is through corneal transplants, but it also can happen with dermatografts or contaminated surgical instruments. Pharmaceuticals that can pass this on are contaminated growth hormone medications that are harvested from the cadavers of human pituitary glands. And again, as all the others, they're fatal, and the survival is about 7 months after the onset of symptoms. So variant CJD statistics are, it's contracted by ingesting contaminated meat. It was first described in the United Kingdom around 1995 or 1994. The CDC reports that 232 cases worldwide as of 2019. It's not real prevalent, but they are tracking them. Again, it's fatal. Everyone who has contracted this has died. Most of the cases are in the UK and only 4 in the US, and those 4 did have connections with the UK where they might have traveled there. It tends to affect younger individuals. The median age is 28, and the range is between 11 to age 74. What you're going to see on histology is cortical plaques and a spongiform halo. So it does poke holes in the CNS tissue, but around those holes, it's going to form a halo around another type of structure, either around a capillary or a neuron or some other kind of blood cell. So that's the key thing with variant CJD. You're going to see a halo around the spongiform damage. So it's caused by consuming contaminated meat that was from contaminated beef cattle, where the cows were fed infected meat. Now, cows are not carnivores and should not be eating meat, particularly their own kind. So they get this bovine spongiform encephalopathy, also known as mad cow's disease. Now, if a cow has this disease, you cannot get it from drinking their milk or cheese or any kind of dairy product from an infected cow. You have to actually consume the meat of an infected cow to contract variant CJD. Fatal familial insomnia is a genetic autosomal dominant disorder. There is a mutation of a prion protein gene known as PRNP. The onset is about midlife, and it is also fatal, and death within 12 to 18 months of the first symptoms. It mainly affects the thalamus. It is a bit different. You don't see widespread spongiform at all with these individuals with this condition. You're just going to see damage to the neurons and glia cells of the thalamus. And the thalamus plays a role in the sleep-wake cycle that we talked about in neuro and muscle physiology. The signs and symptoms are eventually the patient will not be able to sleep. They'll have insomnia, ergo that's why it's called fatal familial insomnia. They'll wake up one day and never be able to go back to sleep. Well, if somebody doesn't have sleep, and we did study sleep, how important that is, we studied that in neuromuscle physiology. Lack of sleep leads to stupor, ataxia, autonomic disturbances. It's going to affect their heart rate, respiration, digestion, and eventually they die from not ever being able to sleep. The image you see on the right of two individuals, these are twins from Australia, and they are currently participating in research projects to find a cure for fatal familial insomnia. They have it in their family. They haven't contracted it yet. They're young, in their 30s, and hopefully they don't contract it. But if they do, they're participating in this research to help find a cure for it. I do have a video that's, I think it's only like 2 minutes, that does talk a little bit about their journey and this research project. FFI statistics are, you'll see abnormal prion buildup in the thalamus. The CNS damage is reasonably confined to the thalamus. You don't really see it anywhere else. It'll destroy those neurons, the thalamic neurons and glia cells. And the histology will show neuronal loss and reactive gliosis in the anterior ventral and dorsal medial nuclei of the thalamus. But it won't show any spongiform pathology.